Good afternoon, everyone. It is Sunday, October 24th, year 2021. It's 2:37 p.m. and hope you are all doing well. Having a beautiful day here in New England. My name is Mark Kulak, Kulacz. It's my little show, Houstonic Live. And I like to do programs on Sundays, and I'm going to do an earlier one today. Matter of fact, we are doing an earlier one today. And in this episode, which is number 293 overall, we're going to review and celebrate the life of a Dr. Francis Plummer, who we've spoken about quite extensively on this show before. This show is Dr. Francis Plummer, past Canadian microbiologist, immunologist, who passed away in January or early 2020. Now, because of the nature of the pandemic and many questions regarding the circumstances of what led to this, yes, there are, and we're very open about questions regarding the timing of Dr. Francis Plummer's passing. That said, these terrible things do happen, but this isn't really about that. Our point is not to focus on that. The point is to focus on this, I would say, rather remarkable doctor. There's many doctors who we, in our research, we stumble across, and it's just hard to tell what their motivations are. And it's very easy to start to focus in on a lot of the questionable actions. And to be quite honest, Dr. Francis Plummer is one of the most exceptional people that I've been able to find in this. And there's several videos which I have very low view counts on YouTube. And I'd like to share these with you. I already have local copies, but I want to share them in the public record. Just so that you yourself can see how other people spoke of Dr. Francis Plummer so highly. There is about 20-minute or so presentation he did at a TED Talk, which was captured. The video resolution and the frame rate is exceptionally poor, but the message will get through. So we may replay that. And this is really just about who Francis Plummer was, what he was thinking, his work, etc. It's really up to his family if they had any questions to pursue anything beyond that, and not for us. So with that said, what we're going to do is we're just going to quickly read the Wikipedia, which we have captured on our HoustonicITS.com site. We'll then flip back and forth between a few of these highlight videos of him, or some of them are older videos that captured some of the research that he had been doing. As you know, he look, we all have our imperfections. He did struggle with alcohol addiction for a while, but he did not hide from that. As a matter of fact, he used it as an opportunity to showcase some of the advances in science with respect to neurology, with respect to implants, which can be used to help people to control certain addictions, which are quite honestly beyond most people's ability to correct. Because addiction is at some point it does turn into, for all intents and purposes, a physical injury, which does require additional means to assist with that. But again, let's focus on who he is. We have a nice little cover on him, which we stole from the BBC for this episode here. Let's see if I can find a nice. And again, although I've researched the topic extensively, I've not put a lot of work into making this particular episode. I do have the comments on. I'll open them up in a few minutes. I hope you guys are all well. And without any further ado, let us just, what we're going to do, we're just going to jump right into it, everybody, here. We're going to do this and this and this. So this was a 2015 video made. So again, this is 5 years before he passed. This is from the Canadian Science and Technology Museum All-Stars. Links are on houstonic.its.com. Just go to the P's, find the plumber. Funny pun there. And then you'll find the page for him.
Meet Dr. Frank Plummer. He's a renowned HIV researcher and the former head of the National Microbiology Lab of Canada. We asked him about his start at the lab, the importance of the facility, and the challenges he faced there.
I became scientific director of the National Microbiology Lab, which is part of the Public Health Agency of Canada, in 1980. And I left Kenya and moved back to Winnipeg. I continued the research in Kenya, but I thought I needed a more sophisticated laboratory approach in order to get where I wanted to go, rather than working in the field in Kenya. It was an interesting challenge. The lab, when I took it over, was very new, and it had relocated from Ottawa to Winnipeg, and only I think something like 25 people moved from Ottawa to Winnipeg. So the lab was kind of underutilized, underpopulated. And I had a big building job to do, which was, a unique challenge and a very exciting opportunity. And the lab was responsible for all aspects of public health related to infectious diseases, from bacteria and foodborne problems to deadly viruses like Ebola. It has the only level 4 program in Canada and one of the very few in the world. So we became the focal point for any infectious disease outbreak, especially when the agent's unknown. The first big thing that we dealt with was SARS in 2003, when this new virus that had never been seen in humans before suddenly appeared first in China, and then by the time we had recognized that there was something of this nature going on in China, it was in Toronto and Vancouver. This severe respiratory illness was undiagnosable using tests for agents that we knew about. So we pretty quickly knew that this is something totally new and had to take a different approach. So it was first isolated, the SARS virus was first isolated by the Center for Disease Control in the US, and then we isolated it shortly after that, like a day or two later. Then we set off on a race, basically, to sequence this virus, to sequence the whole genome. And in collaboration with the University of British Columbia Center for Genome Sciences, we were the first, and Canada was the first, to sequence this. And it was a very, very exciting time. Very exciting time to discover this virus and figure out what to do about it and develop new diagnostic tools. And then the scientific race to sequence the virus was also very exciting. And after that, we went through a number of scares related to influenza, which is, as many people have heard, pandemic of influenza is one of the most feared thing in infectious diseases. The 1918 flu killed many millions of people around the world. We had a pretty severe test of that in 2009 when this H1N1 influenza virus, which was totally new, emerged first in California, and then had a big outbreak in Mexico City. And we helped the Mexicans with their response. But by the time we knew it was in Mexico, it was already in Canada. There were cases in Nova Scotia, so it happened just bang, 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 bang. And it was basically, we were the ones that more or less found the pandemic, because it had already been reported in the US, but we found it in Mexico, and we found it in Canada, and then we knew it was pretty much going to be everywhere. In terms of preparedness for this kind of thing is extremely important. You need to have the scientific capability to respond to it, to make new diagnostic tests, to test new therapies, to make vaccines, hopefully. And I think Canada is very well prepared. The National Microbiology Lab is one of the best in the world, if not the best in the world. To illustrate how important the lab is in sort of the national and global response to these kind of threats, we've done 3 very innovative things that come out of the lab, not by me, but by others. So the first innovation was to develop mobile laboratories, sort of lab in a suitcase, or lab in 12 suitcases, really, that can take the lab to the patient rather than take a specimen to a lab somewhere in the United States or Canada. And that has been incredibly important in the responding to these outbreaks of viral hemorrhagic fever, Ebola and Marburg, because that shortens the time it takes to distinguish who has Ebola, which then you can take appropriate action to isolate them and treat them, or has something else, and who would then be separated from those that have Ebola. The second innovation was the development of a vaccine for Ebola, which I think, and most others think, that it's the most promising vaccine candidate out there. And it was developed using a virus called vesicular stomatitis virus, which is an infection of cows. It can infect people, but doesn't really cause any problems. And putting an Ebola gene in there so that it fools the body into thinking they've seen Ebola, and they develop appropriate immune response to that Ebola protein. And it protects monkeys from challenge 100% of the time if they're vaccinated before they're challenged with Ebola. And you can even have an effect post-exposure. So somebody gets exposed to Ebola, you can still up to or animals get exposed to Ebola. Even up to 3 days later, there's still a very significant effect. So it can be used as kind of a therapy as well. The third innovation was something called ZMAP or ZMAP, which is a cocktail of 3 monoclonal antibodies that were put together at the National Microbiology Lab by a very creative scientist called Gary Kobinger. Two of the antibodies that are in this cocktail were produced at the National Microbiology Lab, and the other was produced at the U.S. Army Institute for Infectious Disease Research in Washington. This cocktail is very effective in preventing Ebola infection in animal models and in treating it in animal models. And it has been used a few times in people, most notably during this current Ebola outbreak when an American physician and an American nurse got Ebola as they were looking after patients in Sierra Leone. And they received this treatment, and from what I've heard and seen, it basically saved their lives. So there's a big rush on now to produce lots of this ZMAP to respond to the current outbreak of Ebola in West Africa.
So just a couple of notes on that. As you know, the Dr. Kobinger was a famous doctor that he is a very famous doctor in Canada, worldwide actually. Who was his peer? It was Dr. Shi, who was a peer of his in the Winnipeg lab. She was arrested on accusations of being a Chinese spy. Dr. Plummer had been working with her for probably almost since the year 2000. She was a big person, also involved in the development of the Ebola vaccines, as well as some other people in modern times. But he hit the big outbreak. The initial story he talked about was SARS 2003, and yes, Dr. Plummer was involved in the initial sequencing of that. We've talked about before. There was some disputes between the United States and Canada regarding the nature of what SARS was and wasn't, and some of the origins. Excuse me, you can capture the show. You can go back and watch the shows on that. We'll probably look at some of those articles in a moment. And I think there's a part 2 to that particular video. Before we go there, let's just review. So now that you've seen him. So the Wikipedia that we've captured back in about a year or so ago, Francis Allen Plummer, Dr. Francis Allen Plummer. Let me make this a full page here. And again, the point of today's show is to really be more of just an overall acknowledgement and appreciation for his life and for his career, as opposed to the bigger stories of pandemics and so on. So it's unfortunately, I think this Wikipedia page does not do him any justice. But again, you can't judge much from page length. He was a Canadian scientist, academic, and HIV/AIDS researcher. Dr. Francis Plummer, Alan Plummer, was quote, a recognized specialist in infectious diseases whose work influenced public health policy in Canada and abroad, unquote. He was a distinguished professor emeritus of medicine and medical microbiology at the University of Manitoba and scientific director general, National Microbiology Laboratory. Sometimes you'll see that as abbreviated as NML, Public Health Agency of Canada. From 1984 until 2000, he worked in Nairobi, Kenya, studying sexually transmitted infections. During his time in Kenya, his research described the heterosexual epidemiology of HIV and the role of male circumcision in reducing HIV infection risk in men. While at the National Microbiology Laboratory, he led the Canadian laboratory response to SARS. That's the SARS. Sometimes we refer to it as the SARS 2003 or SARS 2002-2004 outbreak, the H1N1 pandemic, and oversaw the development of the successful VSV EBOV, that's the Ebola vaccine for Ebola viral hemorrhagic fever. Dr. Francis Plummer was working to develop an HIV vaccine at the time of his death and was believed to be the first recipient in North America to receive DBS, or deep brain stimulation, to treat alcohol use disorder in an experimental trial. Honors. In 2006, he was made an Officer of the Order of Canada. In 2009, he was made a member of the Order of Manitoba in recognition for being, quote, an internationally renowned physician, scientist, and expert in infectious diseases who has significantly contributed to global health, unquote. In 2004, he was awarded the Killam Prize for AIDS research. In 2014, he was awarded that prize. In 2012, he was awarded the Royal Society of Canada's McLaughlin Medal, awarded for, quote, for important research of sustained excellence in medical science, unquote. Now, let's see here. So that was the innovator one. Let's look at here's another tribute. And again, we're just going to flip back and forth between these, right? Here's a tribute to Manitoba's Dr. Frank Plummer that was made in October of 2016. It's made by a celebration of global research.
Over 33 million people on the planet are infected with HIV. Here in Manitoba, cases continue to climb, leaving us with one of the highest per capita HIV rates in the country. But this global epidemic has hit hardest in sub-Saharan Africa, and in countries like Kenya, AIDS remains the leading cause of death. In 1980, when knowledge about HIV and AIDS was still in its infancy, a senior infectious disease fellow by the name of Frank Plummer went to Kenya for a year to study genital ulcer disease. Dr. Alan Ronald, who helped establish one of the first HIV/AIDS investigation clinics in Africa, remembers those early days.
I was trying to get a program started in Nairobi. It was very early days. We didn't have any money and we had very little support systems. And Frank and Carla were vulnerable to being asked to go on this exotic safari to Nairobi. And my idea was that they'd continue the studies that we were doing, trying to understand why so many people in Kenya, particularly the men, had ugly infections of their genitalia. Following from this was his studies of sex workers and how they respond differently when they're exposed to HIV. And some get infected, some don't. Some get infected and they die very quickly. Some live like most of us here do for a long time. And these are all very important initial observations that Frank now has built a huge research program around trying to understand how do some individuals get exposed to HIV, but they don't get infected.
For the next 17 years, Nairobi was home to Dr. Plummer and his family. His work there has led him to become recognized as a global leader in the search to find a way to stop the spread of HIV. Frank Plummer was born and raised in Winnipeg, Manitoba, and received his medical degree from the University of Manitoba. He trained in internal medicine and infectious diseases at the U of M, as well as the University of Southern California, the University of Nairobi, and the U.S. Centers for Disease Control in Atlanta. In 1984, Dr. Plummer joined the University of Manitoba faculty, where he is currently a distinguished professor in medical microbiology and community health sciences.
I was approaching the end of my PhD here in the Level 4 program at the National Microbiology Laboratory, and I asked Frank Plummer if he would mind sitting with me and talking with me a little bit about some of my future prospects in research. So he made the time to sit with me and talk about what my interests were, and he told me a little bit more about what his lab was doing and the directions he would like to see it go in. And he offered me a postdoctoral fellowship position, which I happily accepted and have enjoyed very much working with him and his group.
Dr. Plummer's credo: to appreciate science, you have to do it. The process of discovering something new is wonderful and exciting, and unless you do it, you can't understand it. Dr. Plummer has lived this belief at every step in his career. He has been an advisor and a consultant in demand by international organizations, including the National Academy of Sciences in the U.S., the World Bank, the World Health Organization, and the governments of Lesotho, Kenya, and India. Today, Dr. Plummer is the Chief Scientific Advisor of the Public Health Agency of Canada and Scientific Director General of the National Microbiology Laboratory in Winnipeg. As Scientific Director, Dr. Plummer is in charge of Canada's only Containment Level 4 lab. The lab is a leader in research for several major diseases and was thrust onto the world stage in 2003 when the SARS outbreak hit. Public health researchers around the world work to understand it. It's a time Dr. Plummer described as doing science under intense public scrutiny.
It was a frightening time. We were receiving samples day and night from patients, and we didn't know what we were dealing with. We just knew that it was a very grave situation. Frank Plummer did an incredible job of coordinating the efforts and really identifying what this was that we were dealing with.
The National Microbiology Laboratory made significant contributions to the identification and control of SARS, and Dr. Plummer's actions during the crisis further defined him as a leader. But Dr. Plummer's primary mission remains to stop the spread of HIV and contribute to the development of a vaccine that could save millions of lives around the world. The collaboration that began in Kenya in the 80s continues to flourish and has produced groundbreaking results. Not only have hundreds of scientific papers advanced the research, the project has also brought critical resources to the effort. Many discoveries made through Dr. Plummer's research have been put into practice. He has developed low-cost educational and practical interventions that are saving lives around the globe. Support for Dr. Plummer's research has come.
Dr. Plummer, first of all, he's one of the few doctors that you'll see who really did work extensively in Africa. You'll see some point of criticism I have for many of the other researchers in the HIV area, where they will talk about Africa. They'll have photo ops in Africa, but they really don't live there. Francis Plummer lived there for many, many years. A huge percentage of his life was spent in Africa. And as opposed to a focus on purely, blood testing and vaccination or monitoring, he spent a lot of time consulting with the sex workers in Kenya and some of the other nearby areas, and trying to find out different treatments for what could help them, knowing that the situation was what it was. As well as bringing a lot of his observations back into into the lab, or I should say, into what he was suggesting for policy. Now, I mean, these are obviously can be sensitive topics, talking about the sex trade. But again, he was very pragmatic in realizing it is what it is, and there are many women, many people who were very thankful of his approach, his much more realistic and pragmatic approach, and how much a spread of infection was perhaps mitigated by procedures which he helped define and outline, which were not radical. Anyways, you won't see many pictures of others doing such extensive work in Africa, and Plummer he totally became it, which is makes him just in my mind an absolute standout.
From many fronts over the years. Since 2001, Dr. Plummer has held a Tier 1 Canada Research Chair from the Canadian Institute of Health Research. In 2005, he received a grant from the Bill and Melinda Gates Foundation's Grand Challenges in Global Health Initiatives to continue studies on HIV resistance and work on an HIV vaccine. Your contributions will help advance the battle against HIV/AIDS, as well as the West Nile virus, influenza, and tuberculosis. They will also be used to accelerate collaboration between the infectious diseases community in Winnipeg and the new Hebrew University Institute for Medical Research in Jerusalem.
I think it's going to be very exciting to see what kind of information we glean from this experience. There's so much technical expertise on both sides, and I think it'll be wonderful.
Dr. Plummer recently visited with Dr. Eitan Yefimov, Director of the Institute for Medical Research, to discuss their upcoming collaborative research projects. They are convinced that this sharing of knowledge will make great headway in their research.
There is always a deficiency in the understanding of how basic data can be translated into an effective clinically significant modality. And this deficiency should be filled up by collaborative approaches, where each scientist on each side of the ocean contributes to the understanding of the same particular disease. And I think that through such a collaboration, we may be able to come up with a new idea and a new approach as to how to cope with those diseases which, to the best of my knowledge, are incurable at this.
And what this doctor is saying is true. One of the things which Francis Plummer had observed by working with so many people in Africa was the tendency, when one would think the perception of the prostitutes possibly being areas where there would be the highest levels of HIV. In some cases, he was noticing the exact opposite, and what he was observing was the tendency for some of these sexually transmitted diseases to be to de-attenuate naturally, or in some cases, the worst of the symptoms to be treatable with readily available medicines. Now, he certainly was very much pursuing the advances in vaccine technology, but Plummer did not view it, and that's, of course, where all the money comes in, as we all know. But what really, another thing that makes Dr. Plummer stand out in my mind was that he was not viewing treatment as a vaccine-only endgame. He was viewing overall health. He was viewing what precautions could be taken, what simple methods would help people deal with these potentially untreatable or incurable diseases. And I'm sure that wasn't necessarily a popular mantra. But what he did was you have to work within the system as it is. And the system is being what it is, and that is one which funds vaccine research above all. He managed to have a very long, multi-decade career impacting the lives of many, many people, positively without necessarily viewing the vaccine as the and the only end game. It was always something which was being worked on, but that really wasn't the point. The point was to save lives and improve quality of lives. And that's what that other doctor was saying. He's looking at the full suite of possibilities. And within this system of vaccine and vaccine-only endgame, Plummer had found ways to help people and treat people while at the same time maintaining the whatever was necessary to do to keep the funding going. Very, very complex balance. He is a model, in my mind, a model of how to make a difference from within the system. It is very difficult, and I believe he is one of the greats. He is, or had done one of the greatest jobs at that. And I really want everyone to appreciate that when it's seen. Sometimes it's not obvious. I believe in his case, we can witness it and we can appreciate it and be thankful for that.
Dr. Plummer has received many honors and awards over the years. Most recently, he was awarded the CIHR Michael Smith Prize in Health Research, elected as a Fellow of the Canadian Academy of Health Sciences, and appointed an Officer of the Order of Canada. If you ask him what's most important in his life, he would most likely tell you it's the strong family bond he shares with his wife Carla and their 3 daughters, Marielle, Jamie, and Danica. Tonight, the Canadian Friends of the Hebrew University of Jerusalem Winnipeg chapter celebrate Dr. Frank Plummer's continued commitment and excellence in medical research and are proud to present him with the Scopus Award.
So let's look at I think a rather important couple of articles here. This was from, we've shown this one before. Let me make this one as full screen as I possibly can right now. All right. This was from March 25th of the year 2003. Possible SARS source pinpointed. For those who I've read this to before, I apologize for the repetition. So this, I can't find any copies of this left in the United States press. I suspect it's been erased in many cases, but the Canadian press still had some copies, which I was able to find through Ancestry. A previously unidentified member of a family of viruses that cause cold-like ailments in humans was identified Monday by the United States Centers for Disease Control, CDC, as the leading suspect in the search for the source of SARS. CDC Director Julie Gerberding said in a news conference in Atlanta, quote, "Right now, for us, this is a hypothesis. It is our leading hypothesis," unquote. Gerberding said the CDC scientists have been able to isolate the virus from specimens from some SARS patients and have been able to grow it in culture. I suspect that the samples that they got were from Dr. Carlo Yerbani, whose last request was to make a donation of blood and tissue samples, which could be used to for SARS 2003 research, according to the official narrative. As well, blood samples taken at different times in the course of the illness show at least some patients have no antibodies against the virus early in the disease process but go on to develop them. That suggests the virus is not something they have been carrying for some time, but has mounted a new assault on their immune systems. But the head of Health Canada's National Microbiology Laboratory in Winnipeg said scientists there have been unable to replicate the results that lead the CDC to believe a new coronavirus is behind the mysterious outbreak. Dr. Francis Plummer, scientific director at the laboratory, said, quote, "We've been aware of these results for a couple of days, and as yet, we've not been able to confirm the results," unquote. Our results were negative for coronaviruses, and we continue to find evidence of human metapneumovirus. On Friday, Dr. Francis Plummer announced his team had found a human metapneumovirus, which is a member of the paramyxovirus family in specimens from 6 of 8 patients for whom the lab had samples. Scientists in Hong Kong had named this particular family earlier in the week as the likely causative agent. So he was possibly the first to have successfully isolated it, but you could also see that there was concern and disagreement at the time as to whether or not it was even a coronavirus or not. There were other articles. It was actually mentioned again that this dispute did not get resolved in a weekend. On September 9th, 2003, the New York Times had an article titled "Outbreak That Wasn't a SARS False Alarm." And you could see there's continued disagreement between the types of coronaviruses and the nature of the virus that was causing SARS 2003 between the United States Center for Disease Control and Dr. Francis Plummer. Actually, it went all the way up to the director of the United States Central Intelligence Agency, George J. Tenet. Looks like he was actually taking the pro-CDC side. So this is not just a few scientists discussing matters related to flus and new disease outbreaks which impact a few hundred people. This goes right up to the very top levels of worldwide intelligence agencies, just to really highlight how important this work is. Okay, so let's watch. I know we got some more videos here. Watch one of the celebration videos here of his life. Actually, that was the one regarding brain stimulation. Is this a good one here? Okay, so this was regarding undergoes novel brain stimulation treatment for alcoholism. So he did suffer from alcoholism. He did talk about it, and he went through an experiment to help with that addiction. I think about a year before he passed. You'll observe that he lost a significant amount of body weight during his last year. It was probably because of a radical change in his calorie consumption during that time. So we can play this video here.
October 28th is National Internment Education Day.
1 second.
First national internment operation between 1914 and 1940.
Just have to skip over this advertisement, everyone.
Good evening. We begin tonight with a made-in-Canada medical first that could have life-changing implications. Researchers in Toronto have successfully used electrical brain stimulation to eliminate the urge to drink. Alcohol addiction causes more than 77,000 hospitalizations a year in Canada and more than 4,000 deaths. Now, a renowned microbiologist says the experimental treatment has cured the condition that was ruining his life. CTV's medical specialist Avis Favreau has the exclusive TV details.
Sit down.
Like millions of Canadians, Dr. Frank Plummer used alcohol to relax. Only when he became critically ill did he face the painful truth. He had an alcohol addiction.
I guess I was in denial, you know. It was pretty obvious now that I look back that I had a problem.
He hid it well. Dr. Plummer had an illustrious career as a physician, head of Canada's microbiology lab, on the front lines of the AIDS epidemic and the arrival of swine flu, a life filled with adrenaline and with 20 ounces of whiskey every night.
I used to think about alcohol all the time. Where am I going to get some alcohol? How am I going to get some alcohol?
When his liver failed, he got a liver transplant, but the drinking resumed despite treatments and therapy to stop.
If I didn't get the drinking under control, I was going to die. And I didn't want to die.
So Dr. Plummer became the first in North America to test an entirely new way of stopping alcohol use disorder using deep brain stimulation. Doctors at Sunnybrook Health Sciences implanted 2 electrodes into his brain while Dr. Plummer was awake and talking.
How do you feel there, Frank?
A little bit more awake, maybe.
Those are the contacts that we can
sending electrical signals to override a part of the brain linked to addiction and alcoholism.
This is, in fact, brain-based illness, and there should be no better way to treat a brain-based illness than a brain-based intervention.
A wire that runs down the back of my ear and into my chest here, and it's kind of like a pacemaker.
The electrical pulses are on 24/7, and they stop him from obsessing about the next drink.
It's given me my life back.
Has it had an impact? Oh, it's like night and day.
Doctors say this is still very preliminary, but it offers hope for the most severe cases.
There are encouraging signs that we're having a meaningful impact on their drinking behavior and on their mood as well.
Researchers plan to treat 6 patients with alcohol use disorder, with results expected in over a year. And they're open to treating more people with life-threatening alcohol addiction, Sandy, if they get funding to expand this groundbreaking study.
It's fast.
So that was about a year before he passed. Let's see here. So that was regarding the Travis. Excuse me. That was in 2019. I'm going to read the from CBC News. An obituary for Plummer. This was from February 4th. So it was year 2020. And then we're going to watch another video. I think we're going to watch that TED Talk video again, which is very low quality. But the audio, I think, is sufficient enough. And it really highlights how many people he was able to impact, where his mind was at, as opposed to the a vaccine and vaccine only solution. Again, the only way you can do the first is to actually sell that the latter is the ultimate solution for everything. That's the balance. That's the only way you can get money in this business. It's unfortunate. It shouldn't be that way. We all know that. But sometimes you just have to look at things and accept the reality, and then what can you do to work within that system? So let's read the obituary here. This is going to have to be a little bit smaller. Can I get it fit? No, I'm going to have to make this a full screen. Hold on one second, everybody. As we just celebrate and acknowledge the accomplishments of Dr. Francis Plummer. That's what we are focusing on this show. Not getting into any of the discussions of other related research. And I love this picture of him. In Kenya. You can tell this was probably within the last year before he passed. So Dr. Francis Plummer, age 67, was in Kenya, where he was a keynote speaker at the annual meeting of the University of Nairobi's Collaborative Center for Research and Training in HIV, AIDS, and STIs. Dr. Larry Gelman, who helped set up that meeting, said Plummer collapsed and was taken to hospital in Nairobi, where he was pronounced dead on arrival. No confirmed cause of death yet has been released. Dr. Francis Plummer was born and raised in Winnipeg, where he headed up Canada's National Microbiology Laboratory for several years. He was also involved in an innovative research partnership between the University of Manitoba and the University of Nairobi, established before the world was very aware of HIV/AIDS. Keith Fowke, Fowke, who's a professor in the Medical Microbiology and Infectious Diseases Department at the University of Manitoba, said, quote, he helped to identify a lot of the key factors that are involved in HIV transmission in the early days, unquote. Before that partnership, scientists didn't know HIV could be passed on to women or to babies through breast milk. Fowke added, he developed a lot of the interventions that helped save hundreds of thousands of lives across the world. Frank was definitely a brilliant mind. As a specialist in infectious diseases, Dr. Francis Plummer's work influenced international public health policies. Matthew Gilmore, who is the Director General of the National Microbiology Lab, wrote in an internal email to staff to the Public Health Agency of Canada on Tuesday, quote, Frank dedicated decades of his time to Africa and led the world-changing breakthrough in HIV prevention, unquote. Of course, the most noteworthy breakthrough that he and his team made was the finding of female sex workers who were resistant to HIV. The gift these women have given science in the years since has been unparalleled. Frank had just a joyful reunion with many of them a couple of days before his passing. Another colleague, Dr. Alan Ronald, said Plummer's death is tragic and a loss for science. He said he was so helpful that he was on the path that would end with the discovery of the HIV vaccine, the road he had started down almost 30 years ago, unquote. University of Manitoba's president and vice chancellor, Dr. David Barnard, said Plummer had a great impact on global public health. Barnard said, quote, he was admired and regarded highly by academics and researchers around the world, and his legacy of seeking to develop an HIV vaccine remains one of the landmarks of infectious disease prevention, unquote. As a side note, I must say, it's interesting when you read the obituaries, the hyper-focus that so many have on the vaccine. You can actually, again, I'm getting a little bit off topic here, but you can see the demand and the hyper-focus of that. The first quote is the one which, to me, means the most. He saved hundreds of thousands of lives. Unfortunately, many focus on he was working for a vaccine. Well, perhaps, but he saved hundreds of thousands of lives, even without having created the vaccine. You can see an enormous breakup. Two parallel demands right here in this in health. Really important to focus on that. Dr. Teresa Tam, Canada's chief public health officer, took to Twitter to remember Plummer. Plummer was most recently living in Toronto, where he underwent an experimental brain surgery to fight alcohol addiction. He was believed to be the first person in North America to undergo deep brain stimulation for that purpose. Dr. Gilmore says Frank was bigger than life. His physical presence and intelligence contradicted the fact that he was actually a fairly quiet and shy guy. Science will continue to build on his accomplishments and improve public health in the world for many years to come. Plummer was in Kenya with his wife, Jo Kennelly, and his stepdaughter, Imogen, and the family had just enjoyed time on safari, Dr. Gilmore's memo said. So I've captured other videos. This one here, this was a... let's see, which one was this? This was from 2016. Untold with Dr. Fauci. Actually, this is a longer video. I've played this elsewhere. I don't want this video to be about potential disagreements though between Plummer and Fauci. Though I think that they were skeptical of each other, and we'll just leave it at that for the time being. This is the video I want to play, and again, when it's very choppy, so you get some pretty well, I think even if Plummer was here, he would say it's kind of funny looking pictures of the brain of his head being split because of the low frame rate. But this is a 2011 video that I just want to play in full, and again, it's just to capture it here as part of the public record, if you will. Just want to look at comments real quick. Frank was on a alcohol rampage a year before he died. He had drank a lot. I mean, I'm very open about the fact of my awareness of addiction, and I don't have shame in it. And he did. He had a very serious drinking problem that did cause liver problems. That's true. While the timing is definitely suspicious, I mean, out of respect, that's really for his family to decide if they wanted to do, if there were any other reasons to be concerned. Regardless of that, his accomplishments and his involvements with so many people are noteworthy and should be part of many discussions. I think there can be a lot more understood about the current challenges we face. by really analyzing how he worked and how he spent his time, what he highlighted as accomplishments versus what others may highlight as accomplishments. Don't allow yourself to limit your view of what someone accomplished by their passing. That's really not the point here. Although, in all honesty, it was the timing of his passing that brought many of us awareness of Dr. Francis Plummer's career. But we're not going to allow ourselves to define his accomplishments by questions about that. So I would like to play this 11-minute video. I think it may even be a 20-minute video here. It's just good to hear his if you have a difficult time, if you don't want to hear about some of the perhaps gorier details of sexually transmitted diseases, this may not be a good video to listen to, but it is what he did have to work with. So we're just going to play it, and I'm going to put it on a smaller screen, of course, so that
students with ADHD
the lower frame rate isn't too much of a problem. And again, I appreciate you guys joining and listening to this. We're going to do some more shows in a little bit. We're going to do them on the Live 2 channel. I want this video to be available to stay online without censorship. So we're not going to talk about other topics at this within this video here. Okay, it's a 15-minute video. Thank you.
Well, good morning. I'd like to thank the organizers for asking me to speak here. It's a great privilege to talk to you about our work on trying to understand natural immunity to HIV so that we may...
Audio is very tricky here. I'll continue. You may have to turn up your monitor a little bit. I'm going to do the best I can to improve the quality of the video, the quality of the audio that's on this video. It's not that high, but it's the best that we have to work with right now, unless I can find another copy of this.
Help to inform the development of an HIV vaccine. It's going to be a little bit different kind of talk than you've been hearing earlier today. It's really a very scientific talk. But HIV is a huge problem globally, particularly in sub-Saharan Africa. There are currently about 33 million people living with HIV, the majority of those in sub-Saharan Africa, where it not only has a huge impact on the health of individuals who are infected, but also affects societies, where you have the youngest people in society, the most productive people, dying of this sexually transmitted disease. And in spite of the fact that we have now very effective treatments for HIV, which can prolong life, the pace of new infections being acquired greatly outstrips the rate at which we're putting people on treatment. Only about a quarter of the people that need treatment are receiving it. And I don't think that we're ever going to solve this problem with drugs alone. And we desperately need new prevention strategies and ideally a vaccine for this disease. So this work is carried out, the story starts in a place in Nairobi called Majengo, which is a slum area in the middle of a market, where we set up a clinic for female sex workers in 1985. The reason we set up the clinic was to study immunity to sexually transmitted infections, particularly gonorrhea. But we thought as an interesting side study, and this was at a time when it wasn't recognized that HIV was a huge problem in Africa, thought an interesting side study would be to see that, to determine if there was any HIV in this community. And we enrolled a group of 600 sex workers in our first go at it. And when we tested them for HIV, we found that 2/3 were infected with the virus. This is at a time when it was thought that women weren't very susceptible to HIV, and it was completely a shock that there was this level of HIV infection in this group. So we were interested in trying to figure out why, in the context of the sort of scientific understanding which was wrong, that HIV was a disease of men, how did all these women become infected with HIV? And so we did an epidemiologic study, and one of the main correlates of being HIV positive was the longer a woman was involved in prostitution, the less likely she was to be HIV positive. So that's backwards to what you would normally see. And the only way that could happen is if in this population, some individuals are relatively resistant to infection. And this was at a time when HIV was spreading very rapidly throughout.
Just as a quick interruption here, many of the HIV researchers that I believe are at the top. Another would be a Dr. George Pachenik, actually, who's brother of Steve Pachenik, who's done a lot of work on HIV research and genetics. Will really focus on the fact that there are large groups of people who either have HIV immunity or were developing HIV immunity. And what was it about those populations, which could be studied and extrapolated to help with the fight against it? Many doctors don't talk about that. They'll simply talk about cases, deaths, and the need for a medicine for it. And that's it. But we know health is far more complex than that. And again, there's many things which can't be said here. And I suspect Dr. Francis Plummer also knows that there's things which he can't say, which he's not. But like many diseases, he's focusing on, okay, so why are some people even developing immunity for it? And that should be a part of the solution. So you can see this dichotomy, this breakdown, this breakup of the health industries, if you will, of health policy. And it's an important thing to focus on. Again, I'm really sorry about the ringing in the background. My soundboard's pretty good, but I don't know how to use it well enough to quite filter that out. So I'm sorry. Just doing my best to filter that out. And again, I apologize. It's on the source. This is the only copy I can find of this video.
Kenyan population, ultimately resulting in about 15% of the entire healthy young adult population being infected with HIV. And sex workers were at the center of this outbreak, and also very susceptible to infection, but also important in transmitting the infection onto others, their partners. We put in place interventions to try to reduce HIV, notably using, we had a very rapid increase in the rate of use of condoms in this population, but that didn't stop HIV. And our initial hypothesis that some people were relatively resistant to HIV is really you can't test that in people, and there aren't any appropriate animal models. So what we did is to follow individuals over time. And what we saw is this is what we call a Kaplan-Meier plot showing the fraction of individuals who remain HIV uninfected. So we saw over the first 3 years of study of these HIV negative individuals that about 50% of individuals became infected over 2 years and 60% over about 3 years. But gradually the rate of new infections flattened out and people were less and less and less likely to become HIV infected. And this essentially proved that some individuals were resistant to HIV. We're not the only ones who are finding this. There are populations around the world who appear to be relatively resistant to HIV infection. And that can be couples in which one individual is positive and the other isn't. It's gay men, it's female sex workers, it's children born to HIV-infected mothers. This is an area of great interest because we think, and others think as well, that this is a model of natural immunity. And if we can understand what constitutes natural immunity, really asking two questions, what is protecting these women and why them? Why are they special? We may be able to develop an HIV vaccine. And we've done a lot of work on this. We've been really working on it since 1985 or so. And we know that it's not a result of differing sexual practices or that they have HIV infection that we can't detect by normal tests. We know that certain genetic markers that are important in North American or Caucasian populations are not involved. And everything we have learned points to two things. One is that there seems to be immunity involved, that they have certain types of immune responses to HIV that people who haven't been exposed to the virus wouldn't have, and also that there are familial associations. This phenomena tends to cluster in families, so we think there's probably some genetic basis for it. Am I supposed to point this out? So we've had money over the last 5 years from the Bill and Melinda Gates Foundation for something called Grand Challenges in Global Health. And our project was entitled Immunity, Not Luck, trying to comprehensive studies of the mechanisms of HIV resistance in these highly exposed, uninfected women. But one of the things, you may have a trouble appreciating this, but one of the things we're able to do is look at the gene expression of about 56,000 genes using what's called a microarray. And each of these little rectangles on this slide represents the expression of one gene. And the green indicates that the gene is downregulated compared to controls, and the red indicates it's upregulated compared to controls. What we found was in our resistant women, which are shown here, they have a very, very similar pattern of gene expression to each other, but it's quite different from what we see in individuals who are susceptible to HIV, who are like you and I. So we have, we think, a gene expression phenotype that is a signature for these women, something special about them. And it's kind of reassuring that they all seem to be fairly similar. So it may be that we can figure out what the one thing that is involved in protecting these women is. The one thing seems to be something we're calling immune quiescence. These individuals have lower overall gene expression in cells of the immune system. They also have lower gene expression of genes that are important in HIV replicating. HIV has to hijack the body's cellular machinery in order to replicate. And also lower expression of genes that are involved in cellular immunity. However, they have normal ability to respond to other types of antigens, what we call recall antigens. So if you expose their white blood cells to influenza or other types of infectious agents, they respond like you and I would, but they just have a lower overall baseline. And that's important in that HIV requires activated cells in order to replicate. It doesn't replicate in cells that are quiescent. And so we've developed what we call a two-phase model of HIV resistance. So in a susceptible individual, you have a large number of activated cells that are targets for HIV. The virus is able to infect these cells and replicate. And the body, through its immune system, can't really deal with the infection because it just happens too fast. In these HIV-resistant women, whatever is controlling this, they have fewer activated cells, the fewer cells that the virus can infect, and their immune system is able to deal with the infection. We're very actively trying to understand what is behind this phenomena of IQ, or immune quiescence. And we think if we can understand that, we may be able to make an HIV vaccine or develop topical therapies that may prevent infection. And we identified a number of factors in the female genital tract, and this is meant to be the surface of the vagina, a whole bunch of different things, such as this protein elafin or trypin-2, alpha defensins, SLIPI, that appear to be involved in mediating this phenomena. We don't yet know which is the one that is most important, but they all point to they all fit in very nicely with this whole hypothesis of immune quiescence. One of the ways in which we're studying this phenomena is through something called proteomics. And we have very, very powerful tools now in biology, something called systems biology, where instead of studying one thing, like one protein or one gene, which is what we used to do, say 7 or 8 years ago, you can study all of the proteins in a particular sample and try to understand the interaction. And that's important because you can't understand something as complicated as the relationship between the human body and HIV by studying one protein at a time. And so we use a tool called mass spectrometry to measure all of the different proteins that we find in the genital tract of these resistant women. And this is work that was led by Blake Ball and Adam Bergener from our laboratory. And we found that indeed there are differences in the expression of proteins in the cervical vaginal fluid of these HIV-resistant sex workers compared to controls. And this is a picture of how we measure this. Each little dot on this on these gels represent one protein. And we found that a whole bunch of proteins are different. The proteins that are overexpressed in these women are proteins that involve in dampening the immune response, making the immune system quiescent. So fitting in very nicely with our whole hypothesis of immune quiescence. And the ones that are underexpressed are those that are related to HIV replication. So some of these proteins have been known to be involved in inhibition of HIV previously, but others are totally new. And some of these proteins may be useful as a topical microbicide or used in concert with an HIV vaccine to make people immune to HIV. And we hope to begin to take some of these natural proteins through the process of preclinical development and ultimately into clinical trials. So this two-phase model of HIV resistance, which I showed you before and explained, seems to be really critical in this whole phenomena of resistance to HIV, and also points to the fact that I think strengthens the argument that these individuals are in some way immune to HIV. As I said, this is not the only group in the world that appears to be have different susceptibility to HIV. You have lots of other groups around the world. And about a year and a half ago, we brought the community that studies this phenomena together in Winnipeg. It was in November last year. It was co-hosted by the University of Manitoba and the International Center for Infectious Disease. We had 100 scientists from 5 continents gathered to share information, and ultimately the result of that meeting was a formation of a consortium that to study this phenomena, so we can exchange information, exchange specimens, and kind of come to some common understanding of what is underlying this phenomenon. So this is the largest group that was ever assembled to study correlates of HIV protection, and all in all, there are about 20,000 subjects involved in this consortium. And we have great hope that we'll be able to come to some kind of understanding of what constitutes natural immunity to HIV, what's protecting these individuals, and what's special about them. So I'll stop there with the story not complete. We haven't developed an HIV vaccine, but we believe that this is the path to do that, either a vaccine or some kind of therapeutic that might prevent infection. And maybe in a couple of years I'll come back and finish the story. Thank you.
Denise on the chat said, so Dr. Plummer was interested in natural immunity and genetics, not driven by a push for a vaccine. The summary is yes. And I'll repeat it again. Here's the challenge. The challenge is the power and the money which is being poured and that is available for research on this topic is almost exclusively there. If that person seems to have an end goal of a V or some type of gene therapy, which is usually done through a V. So if you're someone who is really trying to make the world a better place, if you want to help people who are succumbing to this disease, and let's not even get into how it may be spread, you have to work within that system. And I'm trying to highlight, because what we do on this show and other research shows is we're making judgments. We're trying to analyze what are people's motivations, where are they coming from, and almost all of the people that we are researching are at some point or another inside of this machine, and it becomes very difficult, very easy to become very dispirited and to think very negative things about all of the individuals involved. And we'll talk about others in different shows, but in this case, I suspect Dr. Francis Plummer was a rare gem of a person where you could see he had helped, literally saved hundreds of thousands of lives within a machine which wanted him to solve it differently. Now, and that needs to be, you need to take a step back and understand that. And it's appreciation for him. And hopefully it will help us to better assess others who are in this machine right now. That's really the point. For some, perhaps, someday new medicines will be the best preventative. But that hasn't been created yet, and he still saved many lives. Not to say, not to call out too much of a negative about another person, but I don't recall Dr. Fauci, for example, because this isn't about Fauci, saying that he was proud that many lives were saved doing natural methods. It's always about cases, deaths, and we need a V. So I just want to say thank you to Dr. Plummer. We're just going to leave it there because I want this episode to be able to stay online, and for people who just want to see Plummer for the work that he did. He will continue to be a topic of conversation, but again, he was not going to be defined by his passing. Instead, it's his body of work and what he was able to do during that time, and that we should all be pushing ourselves to work at a higher level as well. So thank you for joining. I'm going to be doing another stream within very shortly on the backup channel. Again, I'm definitely going to be deleting all the comments on here because I don't want this to be contaminated, as I know some people may be trying to do right now. So that's it. If you don't join the other channel, have a great day. Thank you for watching this one, and thank you, Dr. Francis Plummer, for your contributions. I'm going to close this out with the with just saying that, and thank you, everyone, and God bless.